I'm Ksenia Blinova from the Division of Applied Regulatory Science at CIDR. Before joining CIDR, I was at CDRH, Center for Medical Devices, and I led regulatory science research across cardiac, neural, and computer modeling, as well as served as technical reviewer and FDA liaison for qualification programs at both CDRH and CIDR. My own research background is in cardiac electrophysiology and, most relevantly to this meeting, assessment of human stem cell-derived cardiomyocytes for prediction of proarrhythmic risk assessment as part of CIPA initiative. Hopefully references to this experience will be helpful in my talk. Here's my disclaimer. So our division's mission is to move cutting-edge science into the regulatory review. Advances in system biology, stem cells, engineering tissues, and computer modeling present new opportunities. Our division has laboratories that work with microphysiological systems across several organ types, including heart, liver, gut, and most recently some women health. Yes, thank you. I was listing the organ system, heart, liver, and some related to women health models. The CIPA initiative combined in vitro and in silico approaches, and we're looking at new ways of creating disease-specific cellular models, especially in rare disease space where clinical testing enrollment can be particularly challenging. Multiple steps are required, though, to translate these new technologies into regulatory use and maintain the same high standards for FDA-regulated products. Drug developers can use new methods in the FDA submissions, but without prior qualification, it adds complexity and uncertainty to the review process. Our qualification programs streamline this, enabling faster, more predictable evaluations. Qualified drug development tools are particularly beneficial for smaller companies with limited resources that are better spent on the medical product development than qualification of new tools. In brief recap of yesterday, FDA's qualification program include drug development tool program and medical device development program. Various types of drug development tools can be qualified by the FDA, including animal models, biomarkers, clinical outcome assessments. The pilot innovative science and technology approaches for new drugs, or iSTAND program, covers everything in between and specifically lists tissue chips or MPS systems as a tool that might be considered for the program, along with, for example, AI-based algorithms. We heard about the 3 stages of the qualification process, including the letter of intent, qualification plan, and full qualification package leading to the qualification. The iSTAND qualification plan and full qualification package templates are currently under development. In the meantime, the biomarker FQP template shown on the left can be followed. There are several key concepts in the qualification documents, including context of use, summary of supporting clinical and non-clinical studies, as well as study design and results, of course. By definition, context of use is a statement that fully and clearly describes the way the tool is to be used in the drug development and regulatory process. The last year meeting focused on the importance of crafting that single winning context of use statement, and I think we made a lot of progress with this specific concept. The only note I would make here is that DDTs are qualified as tool to aid in drug development. So while other uses of the tool may be justified, for example, in clinical decision-making, such context of use is outside of the scope of the program. If there's one thing that I learned from working on MDDT committee at CDRH, it was that it's in your best interest to start with very specific context of use. Here is an example of the COU for the recently approved atrial fibrillation tool from Apple. Many of you probably have it on your wrist right now. I know I do. Look how narrow the approved as a biomarker to this contact of users. It's approved as a biomarker to estimate AFib burden, not other arrhythmias or cardiac abnormalities, and only as a secondary, never primary effectiveness endpoint in clinical trials for cardiac ablation devices. Again, not arrhythmogenic drugs or any other type of medical device. FDA's verdict was that the clinical study provided by Apple was sufficient evidence to demonstrate the MDDT is qualified for the proposed context of use. Accepted emulates letter of intent was mentioned several times yesterday, and I think everyone in this room is very excited about this first CIVM-related successful step. Again, the proposed context of use is relatively narrow. It's for DILI from only small molecule drugs, and it is to be used only in IND submissions to facilitate regulatory decision-making that enables phase 1 clinical trials. I have really enjoyed and had partners discussing their context of use thoughts yesterday, and we can continue this discussion in the breakout sessions. But overall, in my opinion, we made significant progress clarifying how successful COU statement might look like. And really, the question is, what's next? And next to the study, the qualification study, there is no prescribed study design or recommended best practices, as well as guidance document to follow for CIVM qualification as of today. One thing that is clear is that study design would depend on the context of use, and it should provide evidence supporting it in full. If we to design the qualification study today, we would have to rely on general considerations and principles of good science and prior validation and clinical studies. As it was voiced by several speakers yesterday, this is a new area and we're all in it together. So the best study design parallel that I can offer from my own experience is the SIPA myocyte study that we were quite proud of. This study was a large-scale in vitro study to evaluate human-induced pluripotent stem cell-derived cardiomyocytes for the assessment of drug-induced life-threatening interside point arrhythmia risk. This was a huge undertaking with multiple contributors, including drug developing companies, stem cell providers, nonprofit and academic organizations, as well as regulators, including FDA regulatory research scientists. The question of interest was really if iPS cardiomyocytes could predict clinical TDP risk. One of the most important stages of this testing was preparation and coming up with a relevant set of drugs and relevant concentrations. A team of expert clinicians, safety pharmacologists, selected and characterized 28 drugs into low, intermediate, and high TDP risk. Before moving on with multi-site study, we conducted several pilot, sort of proof of concept drug studies in iPS cardiomyocytes and working with cells and platform providers to develop a single consensus protocol. We defined sample inclusion, exclusion criteria, developed predetermined data analysis plan. Variability assessment is very important because you need to demonstrate, say, reproducibility. And in this study, we quantified site-to-site, platform-to-platform, and cell type-induced variabilities. Finally, to decrease bias, blinding plans and blinding drugs distribution plans were developed. Here's the highlights of the study we have arrived at. 10 sites, 5 platforms, 2 human iPS-derived cell lines, 28 drugs tested at 4 concentrations with a minimum of 5-6 replicate wells. So FDA conducted single pilot study confirming feasibility of multi-site effort in 2017 using the data from the multi-site study. On the bottom, a predictive model was built separating drugs with high and intermediate risk of inducing TDP from low-risk drugs. We got good AUC value from ROC curve of 0.87, which is close to excellent performance. Building on this study, we have also published consensus best practice recommendations for iPS assays for pro-arrhythmic risk assessment. And following this effort, iPS cardiomyocyte data started to appear in regulatory drug submissions. So there's a graph on the bottom showing number of iPS submissions in the regulatory pipeline. But as we heard yesterday, the data of that sort is rather sparse, and even when it gets into the FDA submission, it rarely affects the decision-making. So why? Well, of course, this particular study was never envisioned as a DDT. There were no specific context of use developed, so when it becomes a part of submission, reviewers may not know why the data was included. When we did this study, we never made a coordinated effort to work with all the reviewers that may encounter this type of data in submissions. So while we put a lot of effort for evaluating these new technologies, the actual decision makers were largely unaware of the value of this new tool. We were successful in including these alternative methods to the ICH guidance Q&A, but in my opinion, a clear context of use is missing for my sites in pro-rating potential assessment. I'm not aware of any effort to bring these assays through a qualification program. It seems pretty clear, though, that doing so could increase their impact. Just stating the context of use and how they should be used in a regulatory review would help. Which brings me to my final remarks. I can stress enough the importance of bringing stakeholders together early. Today's meeting is great, not only to arrive at some conclusions, but also to build your network across drug developers, FDA scientists, and reviewers, and CAD partners like QMPS, CPATH, you name it. Please take this opportunity, participate, and let us know if you would like to collaborate. After all, we're all working on the same goal here, bringing these new technologies in the regulatory process to improve drug safety and efficacy assessment. Another remark is on the tool adoption. Qualification does not guarantee wide use of the tool. It's important to have all components like reliable cell sources, consistent MPS devices, technical expertise to perform this type of lab work available through the life cycle of the tool. We want to make sure that CDER reviewers are familiar with the tool and its use, so development of reviewer resources should pay off greatly. We want to make sure that the data that comes in follows some consistent format, and the reviewer doesn't have to spend time figuring out what difference would it make if one cell line was replaced with another one, let's say from another donor. On the other side, we don't want the tools to be set in stone. Contrary, we want them to improve with the new science advances. We should learn from the knowledge accumulated from the consistent tool use, and then improve it, refine it, and expand context of use as real life data allows. Again, these advanced models, for them to succeed, collaboration is essential. Whether you're from academia, industry, or government, bring your expertise forward, engage early, and let's work together to establish these tools as a new gold standard. Thank you.